देखती हूँ मैम, कर तो लेंगे। Good afternoon, everyone. It's a pleasure to have you all with us. Welcome again to the international webinar series on compact radiation sources and applications in sciences. We are waiting for the participants to join us. We are also live on YouTube. It's yet another time for talk. This time we will come again with an interesting topic, the use of synchrotron in applied geosciences. The speaker of the day is Dr. Joanna Renshaw. I welcome you all who have joined just now. Welcome again. We are set to go. We are all set to go. We are just waiting for our participants to join us. First, we will have talk, then we'll go for question and answer session. Here are a few guidelines for the smooth conduction of this webinar. All the questions intended for the question and answer session are to be posted in chat box. You have hand raise option. We will start within 5 minutes. Once again, all of you who have joined us, welcome. I think we will start within 5 minutes.
Yes, Dr. Neena, we will start in 5 minutes. Still participants are joining.
Okay, ma'am.
Dr. Monica? Dr. Monica, can you hear me? Monica, ma'am? Monica, ma'am, please unmute yourself. Sanjeev sir, can you hear me? Neena ma'am, you can hear me, I believe.
Yes, you are audible, ma'am.
Monica ma'am, can you hear me? I'll just talk to her and then we will start. Just give me 1 minute. I think, Dr. Nina, we should start.
Okay, ma'am.
I think quite a few participants have joined. I think we should start. In the meantime, the participants will be joining. I think we should start.
Okay, ma'am.
Okay, so you ready for me to start?
We'll be starting, we'll be giving an introduction and then we will start.
Okay, thank you.
Just call you, okay?
Okay. Nature teaches us that tens of billions of light years may have passed, and life in all of its expressions has always been subjected to an incredible combination of matter and radiation. A scintillating afternoon to everyone. On behalf of RKGIT, I, Dr. Neena Sharma, extend a very warm welcome to all the dignitaries, speakers, and all the participants to the last session of International Webinar Series, Compact Radiation Sources and Applications in Sciences, jointly organized by the Department of Applied Science and Humanities, RKGIT, and RKGIT Pharmacy. Synchrotron radiation techniques, that is X-ray, spectroscopy, diffraction, scattering, tomography, play an increasingly important role in research on the geological disposal of radioactive waste. Synchrotron radiation is extremely bright and intense, containing a continuous electromagnetic spectrum of wavelengths, extending from the infrared to hard X-ray region. The unique properties of this radiation allow a variety of analytical techniques. Regarding this, today's topic is the use of synchrotron radiation in applied geosciences. The speaker of the day is Dr. Joanna Renshaw, Associate Professor, Department of Civil and Environmental Engineering, University of Strathclyde, Glasgow, Scotland, UK. We welcome you, ma'am.
Thank you.
Now I request Dr. Poonam Kumar, Head Department of Applied Sciences and Humanities and convener of this webinar series, to introduce the speaker of the day. Over to you, ma'am.
Good afternoon, everyone. Thank you, Dr. Neena. I welcome one and all present here. Before I call Dr. Joanna Renshaw to talk a little brief about her. Dr. Joanna Renshaw is presently working as Associate Professor at the Department of Civil and Environmental Engineering, University of Strathclyde, Glasgow, Scotland. She did MA in Natural Sciences from University of Cambridge in 1995. She received PhD in 2000 from University of Manchester in area of microbiology and radiochemistry. She was a research fellow at the School of Chemistry, University of Manchester from 2000 to 2002, and research associate at School of Earth and Environmental Sciences, University of Manchester, 2003 to 2005. In 2005, she joined the School of Geography, Earth and Environmental Sciences, University of Birmingham as a lecturer in biogeochemistry, and later in 2014, she joined the University of Strathclyde. She is a microbiologist and radiochemist with extensive experience of research into microbial interactions with radionuclides and metals, working at the interface between microbiology and analytical and radiochemistry. Her research interest in development of novel technologies for nuclear decommissioning and waste treatment, microbial transformations of radionuclides and metals, biogeochemistry of radionuclides and metals in groundwater, bioremediation of contaminated land and groundwater. She is mainly focused on understanding how microorganisms can affect the chemistry and transport of radionuclides in the environment. Other recent research projects have investigated the interactions of silver nanoparticle with bacterial biofilms, remediation of petroleum contaminants, and microbial transformations of metals. Currently, she is also a principal investigator of research project on plasma accelerators for nuclear application and material analysis, and also associated with other projects. She is the advisor to Welsh Government as member of Nuclear Task and Finnish Group since 2018. She has supervised 15 PhDs and published more than 32 research papers in reputed journals. Now, it is also a coincidence that today is also International Ozone Day, Ozone Layer Saving Day, and our speaker is also concerned and related to environment. So that's a pleasure, ma'am, having you with us. And over to you, Dr. Jana.
Thank you very much for inviting me. It's a pleasure to be here today. I will start on my talk. I'm going to switch off my video in the hope that my internet connection will cope with this. And I'll start sharing my screen. So can you all see that? Hang on. Can you all see that, my slides?
Yes, we can see your slides.
Okay, great. So originally my colleague Peter Botts was meant to be giving this presentation. Peter has a much more detailed knowledge of X-ray techniques than mine. So mine is more of a general talk, looking at X-ray analytical techniques for applied mineralogy and engineering. So my background is radiochemistry and geomicrobiology. So in this talk, I'm going to give a brief overview of some of the techniques and then look in more detail at some of the projects that we've undertaken to using these kind of techniques. So first, a bit of background about the Department of Civil and Environmental Engineering, the University of Strathclyde. It's a very diverse department. We're split into 3 research groups: the Centre for Ground Engineering and Energy Geosciences, which look at ground engineering, advanced materials science and applications, faults and fluid flow, hydromechanical behaviour of geomaterials, industrial challenges, and non-intrusive geophysical ground investigations. Then we have the Centre for Water, Environment, Sustainability and Public Health, which looks at, amongst other things, soil contamination, remediation and restoration, environmental assessment, waste, energy and the circular economy and international development. And finally, the Centre for Intelligent Infrastructure, which looks at researches intelligent infrastructure systems, smart structures and advanced materials. So first, I'm going to provide an overview of X-rays, and in particular the 2 techniques that we're going to look at examples of today. So X-rays, high energy electromagnetic radiation in the energy range between UV radiation and gamma radiation. And they're generated by the acceleration of electrons, and you can be exploited for a wide range of analytical techniques. I've just got some examples on here: X-ray photoelectron spectroscopy, X-ray fluorescence spectroscopy, X-ray emission spectroscopy, X-ray diffraction, X-ray computed tomography, and X-ray absorption spectroscopy. And these techniques operate at different scales. So, for example, at X-ray absorption spectroscopy, you're looking at an X-ray emission spectroscopy. You're looking at the molecular scale, whereas X-ray computed tomography, you're looking at the nanoscale or micrometer scale. A couple of examples here. So X-ray diffraction, you're looking at crystalline materials, analyzing crystalline materials, and with this you have an incident beam of X-rays which interacts with your target material, and that causes scattering of the X-rays. And the scattered X-rays undergo constructive and destructive interference as a result of this interaction with the material, and this generates a diffraction pattern. And the diffraction pattern generated depends on the nature of the atoms present in your material and their arrangement in the crystal structure. The primary use of this technique is the identification and characterization of compounds based on their diffraction pattern. X-ray fluorescence spectroscopy occurs when you have an inner shell of electron gets excited by incident X-ray photons, and this leads to the release of an X-ray photon from the atom. And the X-ray photon that's released has a specific energy, which is a fingerprint of the atom it's been released from. And so by measuring the energy of the released photon, you can establish the elemental composition of the sample at the point where the X-ray beam has hit the sample. And so this gets used for mapping of the elemental composition of samples. Now today, I'm going to talk in a bit more detail about X-ray absorption spectroscopy and X-ray computed tomography. And then mainly the bulk of my talk is going to be looking at some examples of where we've used these techniques in our research. So first, I'm just going to talk a little bit about synchrotrons as X-ray sources. So a synchrotron is a facility that's designed to produce very intense beams of X-rays, infrared and ultraviolet light. And at the facility, you have electrons fired into the machine where they're accelerated at very high speed by the use of 3 particle accelerators. So the 3 particle accelerators are the linear accelerator, or the LINAC, the booster ring, and the large storage ring. Now, the purposes of the LINAC and the booster ring are to accelerate the electrons so that they're traveling at nearly the speed of light. And from there, the electron beam enters the larger storage ring. Now, this is not the storage ring is not a true ring. It's a polygon made up of 50 straight sections angled together with 50 bending magnets. And as your electron beam travels round that storage ring, the path of the electron beam is bent by these powerful magnets. And when when the beam is bent by the magnets, then the electrons lose energy in the form of light. And this light can be channeled out of the storage ring and into these experimental stations. So as you can see at 0.5 here, this is your light beam coming out from the storage ring. Each beam line has a different section, and this is where you're doing your experiments. You have the optics hutch, where your X-ray is filtered and focused. The experimental hutch, which is where you put your sample, and that's where you're undertaking your experiment. And the control hutch, which is where the scientists control the experiment. So this is just, next is just, sorry. That's the synchrotron in the UK, the Diamond facility down near Oxford. So now I'm going to move on to look at 2 techniques, X-ray computed tomography, and X-ray absorption spectroscopy. So with XCT, you're taking 2-dimensional radiographs to a sample at many different projections around the sample, and then you're computationally reconstructing the 3D image from those projections. So the X-rays leave your source and pass through the sample, there's your sample, to a detector, there. And the sample is rotated and images collected from hundreds or thousands of different angles around your sample. Now, the X-rays interact with the material in your sample, and the flux of the X-rays that is hitting your detector after having passed through the sample is proportional to the bulk density of all the materials and the minerals and the compounds along the X-ray path. And using inverse algorithms, these thousands of individual pictures that you've taken, snapshots of your sample, can be used to recreate a 3D structure, and then you can analyze and manipulate the data to get more detailed information about your sample, and we'll see some of that in the examples that we're working with. So XCT can be undertaken in labs at lab scale. So these are just some basic lab scanners. We have XCT within the University of Strathclyde, or you can do it as synchrotrons as well. And this is just a comparison of the different parameters, the benefits of lab versus synchrotron. So clearly, one clear advantage of the lab is that it's much more easily accessible than going to a synchrotron. Spatial resolution tends to be larger with the laboratory than the synchrotron. Beam energies with the laboratory, you can get between 20 and 320 kilovolts, depending on your system. Synchrotron, it's between 5 and 150 kilovolts, but the flux of the energy, the flux of electrons the flux of X-rays that you get is much higher with the synchrotron. So you're getting a lot more of those photons at whatever particular energy you want. One advantage with the synchrotron is you can get monochromatic light at any energy you want between those 5 and 150 kilovolts. You can tune the energy of your X-rays to a particular energy. So because you have a much higher flux at the synchrotron, the resolution, the time for analysis is much shorter. 1 to 5 minutes is standard, and they're looking at quicker. With laboratory systems, the resolution is, the time it takes to take these samples, these scans, is much higher. So that means that if you're looking at, if you're wanting to understand systems on a quick timescale, then you need to look at synchrotron data, synchrotron system. So now I'm just going to talk briefly about some new equipment that we have at Strathclyde, the GEOX suite of tools for in situ imaging under geological conditions. This is a kit that Catherine Dobson at Strathclyde that leads on. So if you have any further information about it, further requests about it, you suggest contact Catherine. And this is funded through the Natural Environment Research Council in the UK. And this is a suite of equipment to help us do more advanced XCT. So it allows us to undertake not just kind of images of a XCT analyses of a sample, but to be able to do experiments whilst we're analyzing that sample and being able to control different parameters. So we have some furnaces that allow us to go up to 1200 degrees centigrade. So you can look at cooling and heating of your sample whilst you are doing the XCT analysis. We have low temperature as well. Now the advantage of the low temperature is you can have multiple gradient zones for the thermal gradients, and we can also have a controlled atmosphere in there. And that can be, sorry, I've gone backwards. Coupled with other equipment that we've got, which I'll talk about in a second. We have deformation cells, which means that you can put your sample under pressure, under load, whilst you are taking whilst you are doing the XCT analysis, so you can see how it deforms, how it changes, how it fractures, for example, whilst you are putting load onto putting the sample under stress. And finally, we have cells for flow. So this will allow you to do flow through experiments in your on your samples whilst you are undertaking analysis. So if you want to look at, for example, the evolution of a core of, say, sandstone, whilst you're flowing liquid fluid of different chemistries. So applications for this include looking at CT for oil, gas, and carbon capture and storage. The examples there are where you've pumped fluids through a sandstone core and chemicals, minerals have been precipitated in situ, reducing the porosity of your sample. Sorry. So now I'm going to talk briefly about X-ray absorption spectroscopy before I move on to looking at examples of where we use these techniques. So X-ray absorption spectroscopy is a widely used technique for determining the local geometric and/or electronic structure of matter. At the moment, XAS is predominantly undertaken at synchrotrons, but you are starting to get alternatives such as lab-based systems and using other X-ray sources, which I'll talk about briefly at the end. So the X-ray absorption spectroscopy uses synchrotron X-ray source, which is monochromatic, so single energy, very intense. The X-rays are absorbed by the electrons in your sample, and you eject a core electron from an atom. Now, the absorption of the X-rays and the shape of the spectra that you generate from this are determined by the chemical environment around that atom. And you get two forms of information from it. We're called the XANES, the X-ray near edge spectra, and the XAFs, extended X-ray absorption fine structure. So here we just have an example of the raw structure, the raw spectra that you will get out. And this section here, this is your XANES area of your spectrum. And then this area is your XAFs section of your spectrum. And those kind of get separated out and analyzed separately. And they give you different information. So for example, your XANES has, sorry, 1 second, I see a hand's been raised, gives you a fingerprint of the oxidation state of your atoms, of your elements. The XAFs gives you modeling, gives you quantitative modeling of the coordination environment around that element. Second. Hello? Sorry, does someone have a query, or can I carry on? I'll carry on if that's okay. Hang on.
Yes, ma'am, continue.
Okay, sorry, I just saw someone raised a hand and I thought there was a query. Okay, right. So now I'll just introduce you briefly to the two techniques. I'm going to look at a number of projects that we've undertaken in our department. I'm one example from my work at a previous department, where we've used these kind of techniques, the XAS and the XCT in particular. So first, we're looking at colloidal silica grouts for in situ barriers. And this is work done with a number of colleagues, both at Strathclyde and also with colleagues, Dr. Payne and Dr. Kumar at the Australian Nuclear Science and Technology Organisation. So the problem that we were looking at, with this work is legacy waste sites and storage facilities where you've got leaking infrastructure causing contamination to spread into the subsurface. And in particular, we're looking at the nuclear sector. So in the UK, we've had a nuclear industry for over 60 years now. And there's two issues. One is that in the past, waste disposal, the approach to waste disposal was not that rigorous, it's fair to say. And in addition, the second problem is that over time, where you've had concrete facilities for storage or disposal of waste, these facilities have started to degrade, to break down, and you've ended up with leaks. So the examples here are from the Sellafield site in northwest England. This was the site of the world's first commercial nuclear reactor supplying energy, Colloidal, in 1954. And it's safe to say it's the largest and most problematic nuclear site in the UK and probably in Europe. There's a large range of facilities in different stages of decommissioning of varying ages. So the examples, the pictures here are the spent fuel ponds at Sellafield. So these were the facilities where the fuel from the earlier reactors was put for storage. Those are being decommissioned at the moment. And the other particularly problematic area on the site is the Magnox swarf storage silos. So these are facilities that's the outside of the infrastructure, out of the swarf silos. And this, as you can see, this is the decommissioning facility that they started to build around the swarf silos for the purposes of retrieving the waste. That is the top, an old photo of the top of one of the swarf silos. And that's just a diagram of the silo, the inside of the silo. And this is where debris from fuel from the earliest UK reactor program, the Magnox reactors, was stored. Both these in particular are very hazardous and problematic. Now, what we're looking at with things like colloidal silica grout is a means of being able to seal off these leaking facilities to prevent spread of the contamination. Now, for this particular project, we were looking at a site called the Little Forest Legacy Site in Australia, in New South Wales. And this was a low-level disposal trench, disposal trench for low-level waste, and it was typical of many that were built in the time. So we have these in the UK, you have them in the US as well. Another time for low-level waste disposal, the plan was to dig a trench, chuck your waste in and cover it up. And that's the site today. And you can see the trenches, the outline of the trenches there from where they dug the trenches and deposited the waste. Little Forest Legacy Site was operational for low-level waste disposal in the 1960s. They were unlined trenches covered with about 1 metre of soil. There is leaking from the trenches, low levels of radioactivity, including plutonium, strontium, and cesium. Pathways for leaking. So one of the pathways is from what they call bathtubing. So when they get heavy rain events, the trenches fill up and you get leakage along these, you get flow of contaminated contaminated water through these layers here. The other pathway is the trenches go down into a weathered shale region of the bedrock. And this is a very rubbly material. And so you can get flow out through here into the weathered shale. So we're looking at colloidal silica grout as a technology to create injectable, low hydraulic conductivity barriers in environments. So subsurface hydraulic barriers are conventionally created by deploying compacted clays, for example, or geosynthetic clay liners. Now, the problem with these techniques for sealing off contaminated environments is that they require ground excavation. And this limits the sites on which they can be used, because if you've got existing contamination in the environment, barrier installation is not feasible without to excavate, because you've got to excavate the contaminated material. And particularly with nuclear materials, then there's a risk of spreading the contamination. They also can't be deployed on very congested sites, such as the Sellafield site, where you've got a lot of buildings, a lot of hazardous facilities, where you can't really go digging up the facility, digging up the surrounding area. Alternative technologies are grout injections. This is where you inject a grout technology into your subsurface, so like this, to seal off, to form a barrier around your facility. Now, typical grouts that are commonly used are cementitious grouts and chemical grouts. Now, cementitious grouts are problematic when you've got a near surface facility at depths of less than 10 meters. Cementitious grouts are very viscous, thick, they have a large particle size. So for example here, compared to the size of colloidal silica. And that means you need high injection pressures to inject them into your subsurface. And that causes a problem because you can get heave, what's called ground heave, where if you inject high injection pressures cause your ground surface to raise up. Chemical grouts are easy to inject, but they're often expensive and may contain toxic components or interact with the contaminants. So colloidal silica grout is a good alternative. It has a low initial viscosity, a controllable gel time from minutes to days, and I'll explain that in a minute. Excellent penetration due to small particle sizes, so it gets into smaller areas of your subsurface than you can get with traditional cement grouts. It has a very low permeability once the gel is set, and it's non-toxic and a common food additive. So hopefully this will work. So how colloidal silica gel works. You have a suspension of colloidal silica nanoparticles, and you mix it with a salt solution, so something like sodium chloride, calcium chloride. And as you mix the two, hopefully this will work. This is accelerated clearly. The gel sets. It gets thicker and thicker and thicker until it sets as a gel. And as I said, this is a completely non-material. I've actually taken it into my son's school to do some demonstrations with the students there. Now, how quickly this material sets depends on the pH of your solution, the temperature, the concentration of the salt present, the type of salt present, whether it's sodium or calcium, for example, the concentration of your silica nanoparticles. And over time, we've managed to get a very good, the Department of Strathclyde have managed to get a very good understanding of how you can control the gelling of the colloidal silica. So these are just some examples of the lab scale experiments that you undertake. And this is the modeling that we have of the gelling, and you can see it matches very well the gelling in our system. So in order to provide, to look at the feasibility of this in the environment, we needed to look at a number of questions to assess if it's a viable technology for the field. And I'm just going to look at two of those questions that we investigated.
What? Sanjeev ka naam se dalna. Sanjeev ka naam se dalna.
Hello, should I carry on?
Sanjeev sir, please mute yourself.
Okay, I'll carry on. So the first question that one of the questions we're addressing is, what impact does the grout have on the radionuclides in the environment? So if you look at the Little Forest legacy site, we had a leaking trench. And so there are contaminants already in the subsurface surrounding that trench. So we wanted to know, what is the fate of those contaminants? How are they interacting with the minerals and soils present? And what will happen when what would happen when you inject a grout on that contaminant behavior? We focused on strontium as one of the contaminants that was present, and undertake initially strontium absorption experiments with minerals and simulated clays, that simulated clay soil to mimic the environment that we knew is around the the waste trench at Little Forest Legacy site. And then we did grouting and leaching experiments. We took some of our mimicked soil, contaminated it with strontium, and then did grouting and leaching experiments on it. So first, we undertook EXAFs at the Diamond Synchrotron to investigate the fate of our strontium in our clay soil, our mimicked soil. I won't go through the detail of this, but what we did was we looked at, we did EXAFs on strontium. We contacted strontium with the different chemicals we had, the different minerals we had present in our soil. So anatase, the clays, illite, smectite, and kaolinite, and undertook EXAFs to look at their structure. And then we did strontium uptake onto our mixed soil, which was a mix of the anatase, illite, smectite, and kaolinite. And through the analysis, through the comparison of our soil spectra with the single mineral spectras, we could fit the soil sample as fractions of strontium absorbed to anatase, illite, and smectite, and kaolinite. And what this told us was that this strontium speciation was dominated by complexation with tetrahedral silica in the illite smectite structure in the clays. So the next question we wanted to know was, so we know our strontium is stuck to the clays essentially in our soil. What happens when we inject the grout, and if we then flush that grouted soil sample with water to see if it would desorb at all? So these are just the XAS spectra from these experiments. We had a standard of silica, of strontium in a gelled colloidal silica without any soil waste present. The initial soil absorbed strontium before grouting, and then the grouted samples immediately after grouting and after they've been leached for 20 days. And we looked at different kinds of accelerator. And basically the takeaway message from all this, from these comparisons of the X-ray spectra, was that strontium mobility in these systems was controlled by the surface complexation with the clays. So even when we grouted the system, that didn't really change the behavior of the strontium that much. One advantage, though, was that the colloidal silica provided additional surface complexation sites for strontium immobilization. So that meant that by grouting the system, we were actually improving the retention, the immobilization of strontium in the system. Now, the other question we wanted to ask, we looked at, one of the other questions was, what happens to the grout with repeated wetting and drying cycles? Now, when you have hydraulic barriers in near surface applications, the barriers are exposed to natural climatic variations, and you'll get cycles of repeated wetting and drying. And over time, this is known to affect the barrier integrity. It can be compromised. You can get cracking due to desiccation as a consequence of these wetting and drying cycles. And many studies on, for example, traditional barriers, clay barriers, have shown that cracking occurs once you go through multiple cycles of these wetting and drying cycles. So we wanted to look at how wetting and drying cycles would impact our colloidal silica barrier. So we took a kind of worst case scenario, so a kind of exaggerated form of what you would get in the environment. We had sand, which we grouted with the colloidal silica, tested the hydraulic conductivity of it, did X-ray tomography of it, X-ray computer tomography of it. And these are the images that we got, kind of a cross section of it with sand grains and colloidal silica binding the sand grains together. Then we oven dried it. So this is at higher temperatures than you would expect in the normal environment, but it gives us an idea of the worst of a kind of exaggerated upper limit to how it will behave. After oven drying, it was X-ray, it was imaged again, and then water flowed through it to test changes, look for changes in the hydraulic conductivity, and XCT again. And here we have some of the images, the analyses that you, so example of the analyses you can do with the XCT images. So this is just one of the XCT images. Image B shows the available pore space prior to grouting. Image C shows the pore space that was filled by colloidal silica. So these are the white areas, shows you where all the colloidal silica is gone. And this is the pore space filled by air. So these show you where there are actual gaps in the barrier. And through the analyses of these, we developed this conceptual model of what was happening. With the wetting and drying cycles, we did see some nanoscale cracking, an example here, and some mesoscale cracking within the voids. But analysis of the connectivity of these pores that were developing with the wetting and drying cycles indicated that although these cracks were forming, they weren't forming a connected network. And so that meant that there was very little impact on the hydraulic conductivity. With these repeated wetting and drying cycles, the hydraulic conductivity only increased from, let's see, I don't got the details there, sorry. There was little drop in the hydraulic conductivity, which meant that it still performed well, despite these wetting and drying cycles. I'm going to move on to another project or another examples of project, which is looking at, I was asked to include some biology in this, so I included microbes for engineering, looking at biominerals. So what we're looking at with these biomineral technologies that we're developing is you take an organism and depending on, and you give it some nutrients, and depending on the organism that you have and the nutrients you give it, you can make particular kinds of minerals. And we're looking at exploiting those minerals for different purposes. So these are just some examples of some of the projects that we've been looking at over the years. So if you feed it a phosphate source, you can make either trapped radionuclides either in the phosphate form, or we've been looking at generating apatites. One of the projects I'll be talking about is looking at carbonate formation. And if I get time, I'll talk a little bit about the reduction work I did at Manchester. So the first work I'm going to talk about, this is not work that I've done, it's what work that my colleagues at the University of Manchester, University of Strathclyde, sorry, have been leading on, led by Rebecca Lunn, Professor Rebecca Lunn. And this is looking at a process called microbially induced calcite precipitation. Now, this uses an organism called Sporosarcina pasteuri, and you feed this bug urea, and it breaks the urea down into ammonia and carbon dioxide. And this leads to an increase in pH. And when you have calcium present, you precipitate calcite. Now, this is a naturally forming mineral that occurs in rocks. There's a naturally occurring calcite vein. And there's evidence, and it behaves like a grout, and it can greatly reduce permeability and be very long lasting. So there's interest in using microbially induced calcite precipitation to develop a kind of alternative grouting treatment. So here we have some images of the calcite precipitation, SEM images, the calcite precipitation formed, and you can see these tiny little, these are the microbes kind of embedded in it. And this is from another researcher, Van Passem, who's been investigating MICP for ground stabilization. So we've been looking at MICP for a range of technologies, creating in situ barriers for contaminated groundwater flow, look at sealing subsurface reservoirs, for example, for CO2 sequestration, and looking at the repair of building materials such as concrete. That's a project that's ongoing at the moment, and other building materials. So the example here, so these are, this is degraded concrete at the Devonport dockyards in the UK. We have an ongoing project looking at MICP to repair concrete infrastructure in situ. And this is from a project looking at ancient monuments. So this is an example of a sandstone that was very poor integrity. And what we saw was an significant increase in the strength of the material after repeated MICP treatments. So the example I'm going to look at is using MICP as a biomortar for building repair. This is work done by James Minto at University of Strathclyde. in collaboration with researchers at Tsinghua University in Beijing. And they were investigating the potential for using MICP to repair highly degraded marble structures. So in James's experiments, he took loose marble chips and did MICP in the chip column to bind the materials together to consolidate the chips into a solid lump. And this is what you see here. This is experimental setup. Then XCT was undertaken to investigate further, to look at the extent of the MICP within this column of marble chips. And what you can see here is a kind of slice through the column and the average X-ray attenuation down the column. So the more orangey colors are areas of higher attenuation, indicating more dense material. So you've got your chips and your calcite precipitated there. Where you've got kind of more purples and darker colors, that indicates less density. So you've got your pockets of air and the fluid that's flowing through the system. So there's been less MICP there. And what you can see is, not surprisingly, is most of the MICP near the top of the column. This is the 3D reconstruction of the column before and after MICP. This is the flow direction, and this is the porosity. And so you're getting a decrease in porosity as you get calcite precipitation nearer the top of the column. So I'm just looking at my notes. So we were saying that the calcite was precipitated within the column. Other experiments showed looked at the mechanical properties of this and showed that it improved the strength of it. This has implications for how you might want to implement this. So if you just flow it in one direction, then you will get localized reduction in the porosity. So for implementation, you might want to look at, as you saw on the previous slide, no, sorry, 2 things back, about looking at it from different directions or flowing into the center and out into the core like that. This project, I've not really got the results to present yes on this, but it is something that we are looking at, and I just like the project. So this is looking at developing pre-treatment of concrete surfaces to limit contamination. So the problem that you have with concrete infrastructure in, for example, nuclear environments, is you get your contaminants, your radionuclides seeping into the concrete surface. And then when you come to take that facility, decommission that facility, you need to remove that concrete contamination. And for radioactive contamination, you want to try and contain that contamination as much as possible. And the problem with that, if you've got contaminated concrete surface, is that the only way to remove it is through scabbling, for example, which is expensive and messy and very risky for the spread of the contamination. So we were looking at developing a pre-treating concrete to create a kind of decommissionable concrete. You pre-treat the concrete and you do microbial precipitation, you create an apatite layer so on the surface. Apatites are calcium phosphate minerals, but they have a wide variety of structures and they have a high absorption capacity for trapping radionuclides. And we were designing this layer so that, this is ongoing research, so that it could easily be removed once you had finished, once you needed to decommission the facility. So for example, you could have we were looking at whether you could have a metal grid between your concrete and your apatite layer. And if you apply current to expand that metal grid, you could pop off your layer. So the idea is that you significantly reduce the radionuclide penetration into the bulk concrete. These are some images just from previous work, not XCT. So this is confocal microscopy of the apatite and bacterial layer on the surface. And this is SEM of the layer. And we do actually have beam time for this coming up at the synchrotron. We were due to do this in March, and then everything got shut down. The diamond synchrotron is now operational again, and we do have some beam time coming up. It will be remote though. So usually in the past, you get to go down to Diamond and run the experiments with help from the experts at Diamond yourself. Now we're just, for the purposes of because of the ongoing pandemic, we are sending up, our samples will get couriered down and they will conduct the experiments for us. So if, how am I doing for time? Do you want me to pause there? Because, or I can go through the next example. Actually, I'll skip this. So I'll skip through this briefly. This is work I undertook at Manchester. We were looking at how microbes can change the oxidation state of uranium, and this impacts on its behavior in the environment, its mobility in the environment. So just the example here, most stable form of uranium in the environment is uranium-6, and that's this yellow solution here, and that's highly soluble and mobile environment. But this organism can reduce uranium-6 to uranium-4 as uraninite, and that's a black precipitation. Hello?
Continue, please. Continue.
Yeah, sorry. So I'll skip through these quickly and get onto the, because not time for questions. We were investigating the mechanism of U6 reduction. And what we found was that we undertook XF studies of the U6 reduction process. So I took samples and flash froze them in liquid nitrogen, and then they were taken to diamond synchrotron. And what we found was we analyzed the XF spectra to look at the distances between the uranium and the oxygen around the closest oxygen to the uranium metal center. And what you see is a lengthening of this bond length between uranium and the oxygen, depending on the oxidation state of uranium. So when you've got U6, you have the shortest bond length. You have this uranium-5 species, which is not very stable. It tends to disproportionate. That's slightly longer. And when you've got uraninite formed, you have the longest bond length. So what we were able to do is, from analysis of these XF spectra taken over a period of time during the experiment, so from T = 0 to 4 to 24 hours, we could look at the evolution of the uranium species over time. So you started off with predominantly all uranium-6. By 4 hours, we had a lot of uranium-5 present. And then over time, I'm just going to skip back a minute. So by 6 hours, a lot of uranium-5 present. Uranium-5 is not stable in the environment, and it's what a chemical process called disproportionation, where it forms uranium-4 and uranium-6. Now, why does this matter? Well, some of the other actinides, in particular neptunium in the environment, is present as neptunium-5. And that suggests that our bug, our Geobacter, is selective uranium-6 over for +6 actinides over +5 actinides. And that did prove to be the case. When we investigated neptunium-5 with uranium-6, with Geobacter, sorry, and neptunium-5 was not reduced by Geobacter. So it means that if you have an environment where you've got mixed contaminants of uranium and other actinides such as neptunium, your uranium could be immobilized, but your neptunium wouldn't be. It would remain mobile in the environment. Now, finally, I'm going to come on to looking at this new project, which is somewhat of a departure for me, because I am not a plasma physicist at all. This is the Panama project, which stands for Plasma Accelerators for Nuclear Applications and Materials Analysis. And this is looking at developing the kind of techniques that you get at synchrotrons, the X-ray analysis techniques that you get at synchrotrons, but using plasma accelerators instead. We have a plasma accelerator, laser plasma accelerator at Strathclyde, and we are developing this facility there. So just a bit of background. So this is joint between my department and the Department of Physics. So in laser plasma accelerators, you have plasma targets that are irradiated with high intensity femtosecond laser pulses. And these, keep a long story short, this causes you to accelerate electrons in your plasma bubble. And that leads to the release of novel high brightness sources of high energy particle beams and radiation pulses. At SCAPA, we've got 2 state-of-the-art laser titanium sapphire lasers, and it generates radiation pulses from infrared to X-rays and gamma rays, and it generates particle beams such as electrons, protons, and light ions, such as alpha particles. At Panama, what we're developing is we're going to exploit these particle beams and radiation pulses that the SCAPA facility, that the laser plasma accelerator can generate, to create a beamline for a range of analytical techniques. So we're looking at X-ray and gamma ray computed tomography, X-ray radiography imaging, and we've got some examples there of this X-ray radiography imaging, X-ray absorption spectroscopy, diffraction, time-resolved diffraction spectroscopy. Very short radiation pulse time gives us the opportunity to do diffraction time-resolved imaging spectroscopy. Now, the really exciting bit for this will be the opportunity to look at imaging radiation damage whilst it happens. So with the laser plasma accelerator, you can split the laser beam and you can create a particle beam and radiation pulses that go onto the same sample at the same time. So you can use your particle beams to create radiation damage on your sample, whilst using, for example, your X-ray pulses to image what is happening. And so we can undertake pump probe experiments on the radiation damage, so combining the particle beams with diffractional spectroscopy and imaging. So this project started last November. So at the moment, we're very much in the stage of buying stuff and building it. But it could be a very exciting development. Ultimately, so this is with Dino Jaroszynski, leads this for the Department of Physics. Ultimately, what his aim would be is to have basically a kind of lab, an X-ray facility, a synchrotron in a box, essentially. If you can create small enough laser plasma accelerators that you can create them in a mobile facility, then you can take them to your site and have these kind of technologies, analytical capabilities available at your site, for example, at nuclear sites for imaging waste barrels. That was all I was going to talk about, I think. Thank you for listening. Now happy to take any questions.
Thank you, ma'am, for quite elaborating and interesting session. It's now time to move on question and answer session. We would be taking questions from both YouTube and Zoom chat box. Now I request Dr. Pavan Kumar, Dr. Vikas Katosh, and Dr. Amit Sharma to attend the audience for question and answer session.
Hello.
Over to you.
Hello, madam.
Yes,
hello.
sir, you are audible.
Hello, Joanna. Pavan is here.
Hi.
Hi. There is one question from Mr. Lokesh Sharma. What is the fundamental difference between ordinary X-ray diffraction and the synchrotron X-ray diffraction? Because in earlier days, X-ray has low energy. In present day, the synchrotron radiation X-ray has large energy. So what is the fundamental difference between the diffraction patterns?
I'm afraid that is beyond the limits of my knowledge of X-ray diffraction. If Peter was here, he would be able to provide you with a detailed knowledge of it. Yes, sorry, I can't go into the details of that. Very sorry.
Okay, doesn't matter. Actually, what is the bandwidth you use for the synchrotron X-ray radiation?
For which one, sorry? For Peter's experiment, for the for the X-ray, for which experiment, sorry?
Pavan sir, ask your question again.
So I didn't quite catch the.
Pavan sir, can you ask your question again?
I'm just pulling up the papers to look at the details.
Hello.
Yes,
hello.
right.
Hello.
Yeah. Yes. Sorry, I didn't quite catch.
Go ahead.
My question is that what is the required bandwidth of synchrotron X-rays for detection of nuclear waste?
It depends on what you are looking at. So, and I am going to pull up my detail. I can't remember off the top of my head, but we have the tables somewhere because we're looking at for the Panama project. We're looking at this for the energies that we need for our X-ray absorption spectroscopy. If you give me a minute, I can tell you. So it depends, for X-ray absorption spectroscopy, it depends on the element of interest. So you want to tailor your X-ray energy for the element, the particular element that you're looking at. So if it is, give me a second.
For a general element, suppose that uranium like as a nuclear waste. So what is the, yeah.
Give me X-ray edge energies. This is what I was looking for, sorry. So for example, so depending on the edge you're looking at, for uranium, the L edge energy is 17.2 kilo electron volts. So that's the energy of the X-ray that you would be needing for focusing on your analyzing your uranium atom and looking at its oxidation state and its coordination environment. Strontium, for example, if you're looking at radioactive isotopes of strontium, 16.1 isotopes. Cesium, the K edge is 36, which is high. One thing we're hoping to do with the Panama facility is, and this is it, so with the current Panama facility, we're going up to energies of around 20 kilo electron volts. We do have a proposal that we've just submitted to see about getting additional equipment that would allow us to extend the analytical capabilities up to 50 kilo electron volts, which would allow us to include problematic radionuclides that you can't currently analyze at those energies. So, for example, iodine, the cesium K edge. Hello. So those are the energies that we're looking at. Panama facility, we're looking at up to 20 kilo electron volts at the moment. And those are kind of the typical energies of synchrotron radiation for XAS, for example. And
okay, thank you. Basically 20 kilo electron volt is required.
Well, you tune the energy to what you're looking at. So you can get up to 20 kilo electron. I'm just looking to see what we looked at the so we looked at this strontium K edge, which was 16, I think 16 point something, wasn't it?
Okay, thank you.
But for the paper the work I presented on the strontium behavior, they were looking at the strontium K edge, which was 16.1 kilo electron volts. So you tune as your X-rays come in, you filter and focus your energy and tune them to that particular energy, and that's what you focus on the sample.
Okay, thank you for wonderful lecture, Joanna.
There's one more question, Pawan sir, from Rahul Soni. If Rahul can ask himself.
Okay,
Rahul Soni, or if he can ask, please unmute him, or otherwise you ask his question.
Rahul Soni, there is a question. Yeah, just a minute, madam. Rahul question. Not Rahul, there is another question from the Neha Ajay. Explain about the X-ray absorption. Hello?
Hello? Yes. Yeah.
Again, the question is explain the X-ray absorption.
The X-ray absorption spectroscopy?
Yeah.
Okay. So, 1 second. I'm going to come back to my notes. If you have, Peter is the expert on this, so he could give you more detailed information. But with X-ray absorption spectroscopy, you are taking an X-ray tuned to a particular photon energy. So, for example, with the strontium work, we had X-rays at the energy 16.1 electron volts. And they excite, you irradiate your sample with those X-rays. And this causes a core electron. So I'm going to pull this up and share my screen again. Hang on, sorry. So the X-ray gets up. So this is your strontium. Oh, sorry. This is. Can you see a slide from my presentation?
Yes, we can see.
Yeah. So this, for example, is your strontium atom, your strontium nucleus. Your incident X-ray excites a core electron, so electron from one of the inner shells around your strontium nucleus, and excites it out, and it emits a photoelectron. And then the absorption of the X-rays and the shape of the absorption spectra comes from the interactions of the absorbed X-rays and the photoelectron that's emitted, and you get these X-ray spectra generated. So I know my explanation is not very good. Peter is the expert on this. The absorbance of the X-rays depends on the energy, and the depends on the energy of the core electron, which depends on the oxidation state of your element of your atom of interest, and also on the coordination environment around your atom of interest. So that was impacts the absorptions of the X-rays and the different energies. And from analyzing these, you can look from looking at the the ZAMES, the X absorption edge spectroscopy, that provides you with information on the oxidation state of the element that you're looking at. Now, for strontium, it's not very interesting because the oxidation state is always going to be +2. For other elements, such as uranium, where you've got multiple oxidation states possible, then that can help you understand what the oxidation state is of your uranium atom. And then from examining the XAFs, the XAFs tells you about the coordination environment around the central atom that you're looking at. And I'm going to skip through to this one. So what we could tell from these was from modeling the Fourier transform spectra from the XAFs, you could look at the radial distance of the between your oxygen atom, your uranium atom, and your oxygen atom. So these are spectral features arising from the interaction of uranium with its nearest neighbor, its nearest oxygen atom, and you can measure the bond distance from that. And these are kind of from the next, these are from the interaction between uranium and 2 uranium atoms, basically uranium-4 and the next uranium-4. I appreciate that is not a very good explanation. Peter would be able to give you a far more detailed explanation of how XAS works, but unfortunately he can't be here today.
Hello, ma'am.
Yes?
Pune, ma'am?
There was one more question.
There is no more question, ma'am.
Just hold on. Ajay Kumar Upadhyay raised hand. Ajay Kumar Upadhyay, can you unmute yourself and ask your question? Mr. Ajay Kumar Upadhyay raised his hand. Pankaj sir, can we unmute him?
Madam, I have unmuted him.
He just raised hand. Ajay Kumar Upadhyay, if you're there and you still want to ask a question, you can ask. Okay, there was one more question in the chat box and then we can end. I'll just see. There was a question in the chat box. There was a question from Rahul Soni. Yes. There's a question from Rahul Soni. How can I generate powder XRD pattern by simulating single crystal X-ray data?
Again, I would refer you to Peter on that. I've not really talked about X-ray diffraction on this. I can send contact details for Peter.
Okay, that's fine. Okay. So I think no questions on YouTube?
No, ma'am. There is no question from YouTube participants.
Yes. Okay, so over to you, Nina. Thank you, ma'am.
Thank you.
Over to you, Nina.
Now we would be ending question and answer session. Thank you, ma'am. Now, I would like to request Dr. Sanjeev Goel, Professor Applied Sciences and Humanities and coordinator of this webinar series for concluding remark and to propose vote of thanks. Over to you, sir.
Okay, am I audible?
Yes, sir.
Okay. Thank you, Dr. Neena Sharma, for consenting me word of giving, concluding address and vote of thanks. A warm and graceful evening to all. As all good things come to an end, so this webinar series too. I, Dr. Sanjeev Goel, Professor Physics and coordinator of the webinar series, take the opportunity and feel it to be my pleasant duty to propose a vote of thanks on behalf of RKGIT Pharmacy and the Department of Applied Sciences and Humanities, RKGIT, Ghaziabad, India. The organization of this webinar series was a joint effort of the RKGIT Pharmacy and of Department of Applied Sciences and Humanities. It was planned with an aim to explore research areas in the field of compact radiation sources and applications. The webinar series started on 14th September with its first lecture by Professor Zulfikar Najmudin, Department of Physics, Imperial College, London, on the subject radiation sources from laser accelerators and applications. The second lecture of the series was on 15th September by Dr. Antoine, Research Fellow with Professors Dino Jarosniski, University of Strathclyde, Glasgow, Scotland, on the subject laser-driven sources in radiotherapy. The third and concluding lecture of the webinar series was held just now on the subject the use of synchrotron radiation in applied geosciences by Dr. Joanna Renshaw. This webinar series was streamed on Zoom and YouTube simultaneously and attended by a good number of audience from all over the world. We are sure that the topics which are covered in this series must have helped the participants to know the recent research areas. Conduction of this webinar series by the Department of Applied Sciences and Humanities was second in a row in just started new session. The first one was from 4th to 26th August 2020, which was a series of 5 lectures on the subject recent advances in science. We are looking for organizing such webinars and FDPs in near future too, which would kindle our thought process leading to the advances in research work. Now I propose the vote of thanks. I express my sincere gratitude to the Speaker of the Day, Dr. Joanna Renshaw, Department of Civil and Environmental Engineering, University of Strathclyde, Glasgow, for accepting our invitation and enlightening us with the knowledge sharing on the subject, the use of synchrotron radiation in applied geosciences. It was an informative and very interactive session indeed. Thank you, ma'am. We wish to listen to you on more such occasions. We are thankful to our Chairman Sri Dinesh Kumar Goel ji, Vice Chairman Sri Akshat Goel ji, Director R&D RKG Group of Institutions Professor Lakshman Prasad sir, and the Director of RKGIT Dr. D.R. Somasekar sir for their support and motivation to organize the webinar series. Thanks to the participants from all over the world for their presence on the Zoom and YouTube and taking keen interest in this webinar series. Our special thanks are to the whole organizing team who was behind the success of the webinar series. The conveners, Professor Monica Sachdev, Principal Pharmacy, and Professor Poonam C. Kumar, HOD Applied Sciences and Humanities. My partner coordinator, Mr. Abhinav Kumar Agrawal, Dean Academics Pharmacy. Co-coordinators, Dr. Vikas Kotoj, Associate Professor, Department of Applied Sciences and Humanities, and Mr. Pankaj Sharma, Assistant Professor, Pharmacy. Dr. Pavan Kumar, Associate Professor Physics, for inviting the internationally renowned speakers to this webinar series. Mr. Santosh Mishra and Dr. Amit Sharma for technical and active support respectively. Dr. Neena Sharma and Dr. Garima Garg for flawless anchoring. Mr. Pavan Pandey for giving the coverage to this webinar series in the leading newspapers. Thanks to all other colleagues of RKGIT Pharmacy and Department of Applied Sciences and Humanities who were directly or indirectly involved in making this webinar series a successful one. Thank you all once again. Thanks a lot.
Thank you, sir. The feedback link has been shared in chat box, and after filling the link, your certificate will be shared on your registered mail ID. End of the story is the new beginning for many others. On this note, let us conclude a remarkable, memorable, and knowledgeable event. Thank you, everyone. Please turn on your video for a snapshot. Everybody, please turn on.
All the team members, please join for a video snapshot.
One more. Okay, ma'am. Done.
One more, Vikas sir has joined.
Yes, ma'am. Done.
Okay, thank you. Thank you, Dr. Neena. Thank you, Dr. Jonna. And thank you, everyone present here. And we have successfully conducted this webinar series for 3 days. I think we will end in, Pankaj sir, how much time should we take?
In just 5 minutes, we will end, madam.
No, we will not end in 5 minutes because today everybody has to fill the feedback form and get the certificate.
Okay, madam.
So we have to give time. We have to give some time.
No problem.
So maybe 10-15 minutes we can give and then you can switch it off.
Okay, madam.
Okay, thank you, everybody. Thank you once again, Dr. Jonna, Dr. Sanjeev, Dr. Neena, Dr. Monica. I can see them on the screen. Thank you.
Thank you.
Thank you for inviting me.
Thank you.
Welcome, ma'am.